very useful insights we have received from you. We hope to see you at MycoIndia again and again. Thank you very much.
Yes, for sure. Thank you so much.
Thank you. Now I request Dr. Rajesh Jeewon from University of Mauritius to join us for his presentation. Dr. Rajesh Jeewon, if you are around, please say hi.
Hello, everybody. Can you hear me?
Yes,
Yeah.
we can hear you, Dr. Jeevan. Let me just introduce you to this audience. Ladies and gentlemen, Dr. Rajesh Jeevan was my PhD advisor at the University of Hong Kong. About Dr. Rajesh Jeevan's educational background, he did his master's from Mauritius. Then he joined Dr. Kevin White's lab at the University of Hong Kong. He worked on the molecular phylogeny of Pestalostiopsis and morphologically similar anamorphic genera. And he published one of the greatest papers in terms of phylogeny of Pestalostiopsis in the MPE. That time, publishing in MPE was very difficult. Molecular phylogenetic and evolution. Then after PhD, for a brief period of time, he went to the UK for his postdoc. Then he rejoined the Dr. Kevin Knight's team at Hong Kong. And he actually spent lots of time there, maybe 5 to 8 years there, I guess. And he has supervised, along with Dr. Hyde, many people, including me. And I'm very grateful to Dr. Jeevan for helping me, especially with the writing part. He was very, very helpful when it comes to writing. I think because of that, probably I ended up founding 2 journals today. Thank you very much. And other than that, he has supervised so many people from Southeast Asia. And Rajesh Jeevan, and he was the editor-in-chief of MycoSphere Journal, and he's one of the senior associate editors of MycoIndia Journal of Indian Fungi. Thank you, Dr. Jeevan, for all the contributions you have made to my career and also to the field of mycology. In this presentation, I will talk about the importance of fungal taxonomy and diversity. I'm sure it will be loaded with lots of insights and knowledge. Thank you so much. Over to you, Dr. Jeevan.
Thank you, Dr. Shenoy, for this nice introduction. I hope everybody can hear me. Maybe what I will do is I will just share my screen. Can you see my screen?
I think it's getting loaded. I think yes, we can see now.
Okay.
Yeah, I think you could go to the full presentation view, please.
Okay.
Not at the full presentation view, actually. Just before the zoom. Next one, yes.
Okay.
Good afternoon. Sorry to bother you. So it's not at full presentation view. I don't know why.
I'll try. Let me check again.
If you have the PDF copy, you can try the PDF copy.
Let me share the screen again.
Yeah.
Is it far better now?
What has started the loading part? So I think so. It is on the full screen. Can you just move to the next slide, please?
Okay.
No, not yet.
Okay.
Yeah, it's moving. Yes, moving. Thank you so much.
Okay.
Yeah. Yeah.
Okay.
Yeah.
Thank you, Dr. Shenoy. Good afternoon, everybody. Thank Shenoy has given a nice introduction about myself. I will not be taking a lot of time today. I know it's Sunday and a lot of you are rather tired by now, and you've got one more interesting lecture from Dr. Shenoy. So in brief, I'll be talking about the importance of fungal diversity, especially to combat diseases and promote diversity conservation. especially for better environmental health. Many of us think about microbes, but we've got a tendency to think more about bacteria. The world of fungi has been, to a large extent, over the last 30 to 40 years, been overlooked, despite the fact that they do contribute to climate change, they do contribute to nutrient recycling, they do contribute a lot in our industries and even for our biopharmaceutical products as well. I think many of you will know about, the famous penicillin and Louis Pasteur experiment and so on, about how, fungi came into vogue at that point in time. Where I am from, yes, I am from Mauritius, which is basically a tropical island east the coast of Madagascar, and it's about 6 7 hours flight to Delhi or to Bombay. But if you go to where Chennai works, it's about 17 hours. So I stay in a very nice tropical island down here. So, what pops into our mind when we talk about fungi? As I said before, most of us will think about those fungi that we can see with our naked eye. Those mushrooms, for example, those different types of Amanita species, Lentinus species, Polyspora species that we can see in the forest. And while we see that, we can immediately try to understand or to ponder or to think about why are those mushrooms there? Obviously, for a good reason. They are one of the most proficient organisms to break down lignin and cellulose on wood plants and hence contributing to fungal diversity. Yes, ladies and gentlemen, fungi are mushrooms. But surprisingly, most of the fungi that we have not yet discovered are basically existing as microorganisms, and we can't see them with our naked eye. Just to give an idea about how many mycologists nowadays are proceeding with a lot of studies to try to discover more about those fungal organisms. We've got a number of new species that are being discovered every year, and people are quite amazed about how these microorganisms live in the environment. They're almost everywhere. Just to give you an idea, when we talk about the Viagra of the Himalayas, it's cordyceps fungus, and it could be more expensive than gold. And a lot of people, livelihood, especially in Tibet and in the Southeast Asian region, people depend on that for a living. Even when it comes to mushroom, in a lot of places in Yunnan province, which I've just recently visited, there is a lot of people there who are making a living out of fungi. I mean, those people living in rural areas, they go and collect fungi in the forest and they sell it just to live. There are others as well who are exploiting nowadays mushrooms on an industrial scale and making a lot of profit. So, when we talk about fungal diversity, the biodiversity is amazing. Just imagine if we could have one simple substrate on Earth where we don't have the presence of fungi. I would be quite be baffled. Almost every samples that you have, almost every substrate that you examine, fungi are there. You talk about the ocean, even deep inside the ocean, when we'd have those marine organisms, marine sponges, algae, or even in the water column, the presence of fungi on corals as well. There is a number of studies that are basically done in Mauritius, especially on sponges and on algae, and we've discovered a wide range of microorganisms. And based on our NGS sequences that we've analyzed from those marine samples, we were quite bewildered by the types of lineages that are arising from those strands of DNA sequences we are picking from the environment. You talk about freshwater fungi as well. A lot of fungi are actually colonizing our freshwater habitats. You talk about the mud sample, you talk about peats, you talk about worms, you talk about the wood debris, you talk about the leaf litter, everywhere you go. And these habitats are actually harboring a huge number of potentially new species. In the forest, in the canopy, on the leaves, on living plants, they exist as endophytes. And we've done a lot of studies on endophytes as well. And one of the very first study that we came across, and basically one which is very highly cited, was back there in 2007, where we worked out a project and we found that there are actually fungal species existing as endophytes inside the plants. But when the wood starts to decay and when the leaf starts to decay, those endophytes can actually change their lifestyle and start to adopt a saprobic lifestyle. And of course, this means to a certain extent that these microorganisms are involved heavily in nutrient recycling. You talk about the soil. 1 gram of soil could harbor thousands of different species of organisms, especially fungi. Don't be scared, guys. Even in our bedroom, on our pillow, on our mattress, everywhere. You talk about fungi, they are even in our human body. Quite latent, I would say. You talk about insects, we've got a lot of fungi associated with insects. In the extreme environments, in the glaciers, and the hot and arid environment, in hot springs, on mountains everywhere. So these fungi, over the course of evolution, they've been able to actually colonize almost every habitat on Earth. And with time onwards, what has been happening, there has been a number of evolutionary events taking place that have actually given rise to the basic concept that we have about speciation, the rise of new species from existing ones. So we might be wondering about how many now fungal species could there be? We started asking ourselves these questions maybe like 20 years ago, even far ago, but we've never been able up to now to come to even a real estimate about how many species could be lurking in our environment. And this has been on the agenda in a number of meetings, mycology meetings worldwide, where people are still discussing and working out numbers. So how many do we have? I mentioned about speciation before, so we are assuming that there should be a number of new species as well. So we started back there in 1991 with one of the famous mycologists, Hostworth, estimated that possibly there is approximately 1.5 million species. And how he came up with that particular ratio was based on the number of vascular plants that we had. So basically assumed that we had 4 fungi to 1 plant, and hence he came with a number of 1.5. But then things started to change in 2005, because we are coming up with novel technologies. We've got, DNA sequencing that are being done on routinely basis nowadays. And O'Brien and his colleagues came up with the number ranging from 3.5 to 1 to 5.1 million. But then again, with high throughput screening methods where people, are just picking up any samples and, trying to pick DNA from there, and the amount of different fungal sequences which were picked up were actually tallying with supposedly different types of species. And Blackwell in 2000, in 2011, mentioned that possibly there is 5.1 5.1 million species. So now, which is which? This is only up to 5.1, but then again, there has been revision because we've been working on environmental DNA samples as well. So there has been revision, and mycologists started to think about it from a more rational perspective, and it started to say that, well, possibly, we have like 2.2 to 3.8, and then again, we've got Wu et al. Who says no, maybe we've got much more, maybe 11 to 13 million species. But the latest paper that we had estimates about 6.2 million species. Now, surprisingly enough, okay, we've got only about 150,000 species which have been formally described. So now, if we are estimating, let's say even about, let's say the least that we can estimate about 1.5 million species. So we are actually less than, let's say, about 10% of our species currently being described. So the 90% is still there in the environment, awaiting discovery. Now, where are the missing fungi? Are they on land, or are they in the ocean, or are they maybe in the freshwater environments? There is a nice paper. We've worked it out together with Hyde and our group back there in Thailand. And given that Thailand is a fungal diversity hotspot, there has been a number of diversity studies and taxonomic studies that have been done in that region. And surprisingly, they assume that there could be up to 90% of species that could potentially be new in those environments. And even those tropical hosts or plants are not well studied. And their analyses reveal that there could possibly be a large number of new species there that need to be explored. So, could the missing fungi be actually found in many tropical species or many subtropical species? And should we go ahead and do extensive taxonomy and taxonomic work to be able to discover more of those fungi? Just to give you an idea, there has been a various work that has been done in China as well, especially in higher fungi. When I say higher fungi, I'm referring mostly to those, macrofungi, those ones that you can see, those mushrooms in Yunnan. And people are discovering, those Chinese scientists are discovering a number of new species that have never been documented before. Okay. They've documented about 104. They mention about 104, or 1,104,000 species, fungal species. And there could be about 6,000 fungal species that are known there. But this is about higher fungi. And based on our experience, we would presume that the number of species for microfungi must actually be higher. There are also other works that has been done in China, for example, on freshwater fungi. And even with freshwater fungi, it's quite surprising because they are discovering a number of asexual fungi there, especially those on wood and on leaf litter. So what all these studies are telling us to a certain extent that there is a need for better strategies about how we go ahead and collect, isolate, and identify those species there. One thing that we should be aware is that those freshwater species actually could be producing a number of important metabolites that could be very important in terms of the pharmaceutical industry or in terms of human health as well. Oh, the freshwater species there to help us uncover novel secondary metabolites? Yes, possibly. There is a need to go ahead and try to discover those fungal genomes and do more work on the fungal genomics to try to understand about how these fungi are living there and what could potentially be their ecological roles in those different environments. There are a lot of species as well only in Yunnan province. I'm taking the example of Yunnan province is because that particular province in China has been claimed as the kingdom of plants, and a few of the studies that we've been doing there have revealed a number of new species of microorganisms. Now, living alone, wood, soil sample, freshwater species. There are a number of species that are associated with animals as well. And over the past 10 years, one of my colleagues, Dr. Mo Timer from Quming Institute of Botany in Yunnan province, China, have described over 1,000 new species, especially when it comes to mushroom. And they tell you that for every 5 species they encounter, one could potentially be new to science. So, could also uh, could it be possible that Yunnan is a place which holds the answer for where are those missing fungi? With more discoveries, are we getting a better idea about how many species are there? These are questions that are left to be answered. Now, why is there a need for an inventory of those fungi? Because, as I said before, they are very important. They cause diseases. They are here for recycling of nutrients. Okay, they are being exploited to a certain extent in many types of different industries. But there are problems. Why? Because it's very difficult to detect the presence of those microfungi. Even if we detect those microfungi sometimes and examine them under the microscope, because of morphological overlap, because of cultural variation, it's very difficult to basically sometimes identify a particular species correctly. Our sampling protocols that we use sometimes does not make it possible for us to recover all the different fungi. You can imagine you take a leaf surface and you're trying to isolate and divide from it. I mean, only one single leaf, you could be coming up with at least 20 to 25, let's say, different, operational taxonomic units or possibly supposedly different species. Now, do you think in a particular study our sampling protocols will allow us, for example, to work in a very systematic way on soil sample, in a freshwater sample, to be able to recover all those fungi? It's not actually possible. The other problem that we have is actually about basic species concepts. So far, we've got a lot of different species concepts that have been proposed, okay? But mycologists are still in a dilemma about what exactly defines a particular species. And this has also been bothering me a lot over the last 10 years. And when I found that there is a lot of people who are just, identifying and, proposing new species based on how they are doing laboratory work, I actually wrote a paper which we published in Mycosphere in 2016, just to lay out the recommendations a little bit and about the precautions that people should take before they establish new species. And I've got one or two slides there that I will actually capitalize on. The other problem is fungi exist in two stages, sexual and asexual. What I mean in here is that the same fungi can exhibit totally different morphology. The one in the sexual state could be totally different from the one in the asexual state. So let's say if you are a mycologist and you are collecting a particular fungal sample and bringing it back to the lab, and it's existing in its asexual state, so you will try to label it differently as compared to somebody else who has collected the same fungi in its sexual state. But now the problem is a bit resolved, because with the use of DNA technology, with the use of DNA sequencers, we can actually try to link the sexual state and the asexual state together. And this is what we've been doing over the last, back then, 2005 and 6 and so on, especially with Dr. Shenoy's work, where we've designed something very nice with those asexual fungi, and we've tried to integrate them in a proper taxonomic scheme whereby we try to avoid confusion about how to name those asexual fungi. And even as Dr. Shenoy said, that one of the very first paper I published, that was back there in 2001 and 2, where I was almost like the first person to try to integrate a molecular perspective that was integrating asexual fungi in a proper sexual framework, and try to link uh, asexual fungi with sexual fungi and avoid, double naming of species. The other problem that we have as well is that many species cannot be grown in vitro. That's where we see that, DNA sequencing is playing a very important role in terms of picking DNA, fungal DNA, that we cannot grow in the lab. Nowadays, we're also using environmental DNA sequencers. But what is happening with that is that we are getting only uncultured species. We are getting only a particular strand of DNA sequence, and we are labeling them as operational taxonomic units. But do or can this single strand of DNA represent a particular species? Can we name those single strand of DNA, or those fragments of DNA we are picking up from the environmental sample, and deposit the sequence in GeneBank and try to name those species? This is also food for thought, and I've actually written two papers on that. One in Scientific Reports in 2018, where I try to discuss a little bit about what are the precautions and how should we go ahead, and what are the problems arising from those massive kind of work that we're doing from those DNA. Obviously, I would agree that DNA is very important. It's unraveling a lot of mysteries, in terms of taxonomy. But how far can we go in terms of coming up with a proper scheme or a proper framework about associating a particular strand or a particular segment of DNA to a particular species? Okay, there we go. The whole concept nowadays, everybody talks about DNA sequencing. Okay, so what are the novel perspective? Yes, I would say it is important. We've got to try to integrate and go ahead with novel technology. So how do we do it? Basically, just to give you an idea, because I don't know who is basically in the audience in here. So what we do at the very beginning is examine the specimen we are collecting, try to get all the morphological details. We try to grow the culture, but I must understand that in a number of cases, we cannot simply grow the culture. They won't grow at all. But if we can, that would be good. Then we can use the mycelia as starting material. And then we go ahead and extract DNA. As I said before, a number of fungi do not grow in culture. Okay, so we've got to extract DNA directly from specimens. And this is again a nice paper that we published in Mycological Progress about a simple and simplified DNA extraction protocol for, extracting, ascomata and fruiting bodies and try to get decent DNA so that we can go ahead and do molecular work. Once we get our DNA, we go usually for PCR, where we try to fragment, or amplify segments of a particular gene. Once we get the DNA sequences, we try to align them with other sequences. We do blast search results and so on, just to get a putative identity. And once we have it, then we can go ahead, with Mega and try to analyze the DNA sequences in a particular data set, so that if we've got new species or potentially, different species, we can try to put them in a proper taxonomic framework where we can try to infer evolutionary and taxonomic relationships among different species. But again, things that bother me sometimes when I see how some people are just describing species and, maybe establishing new species as well. So at some point in time, I sat down there and I started asking myself, is my fungal species new? Should I describe it as a new species and publish it? Or is it just an artifact of my brain telling me that it is new? So to overcome some of the problems and some of the fallacies or possibly discrepancies I've seen in previous papers, so I come up with this idea about how can we try to avoid problems, especially in taxonomy. Because when we talk about, identifying a particular species, it has got a number of implications. For example, if you misidentify a particular fungus that is causing diseases, then the whole, plant pathology aspects, the whole issues behind, disease management goes wrong. The same happens, let's say, even if you identify a new species, but it's not a new one, or if you even misidentify an existing one, then it has got something to do with biodiversity estimates about how many potential species are potentially colonizing one particular substrate or could be found in that particular environment. So in this particular paper, we came out with 15 recommendations about what people should do when going ahead in describing their species. Okay. So this is just a few that I mentioned in there. So we need to include reference sequences. And there is also a need to check whether those sequences from GeneBank are reliable or not. With my over 20 years experience in working with students and supervising students, I can tell you that there is a number of sequences in GeneBank that are not reliable, and many don't know that these sequences are actually good or bad, and they are just including them in their phylogeny, and this is messing up fungal taxonomy. The second aspect, phylogeny of core genes. Before, when I started, studying, working in the lab back there in the late 1990s, we used to rely mostly on ITS, but things have changed with time now. Even though the ITS has been referred to as the preferred barcode for fungal identification. We are now in a better position to try to understand how different genes behave in different way and what is the importance of including different genes in our analysis so that we can come up with more robust identification. Our taxon sampling is also very important. Right from the start, if you're building up a phylogeny, for instance, and you're trying to infer phylogeny, so you must know how many species you're including in your phylogeny and which species should you include in there as well, because this has implication on your identification. Now, let's say if I'm comparing my species with an existing one in gene bank, and I've got, let's say, if I'm using the ITS, I've got approximately, let's say, 500 base pair. Now, how far does my 500 base pair differs from the existing one in gene bank? And on what basis can I assume or predict that my taxon that I have collected might be different from the other one or might potentially be representing a new species? So I came up with that nucleotide difference. So basically I said there should be, when it comes to the ITS, okay, or any other genes that you use, we should try to stick to the more than 1.5% nucleotide difference. If we have a novel lineage or nucleid that is potentially representing our new species, it should be phylogenetically distinct from related taxa. I've seen a number of new species being described where the cluster with existing species and with 100 bootstrap support. So sometimes it makes me wonder, even the DNA sequences are like 99.5% similar to the other one. So should I establish it as a new species or a different species or the same species? All these are food for thought that many people, especially taxonomists, need to think about in more detail. I would say that there is no problem establishing a new species despite its close connection with one in gene bank, despite its 100% similarity in DNA sequence with an existing species where you have DNA sequences in gene bank. But all these will have to be done in a very scientific way and with strong justification. Okay, so what should we do? I think we should go ahead with an integrated approach. At the very beginning, there's a need to explore all scientific possibilities. To be frank with you guys, there is a possibility of wealth, a lot of wealth out there, in the mycological world. There's a need to scrutinize all these, analyze what's going on, evaluate them as well. Can I just supplement my phenotypic data with genotypic data? Should I go ahead and use a polyphasic approach? Of course, this would be what I would be recommending. Now, how many similar species are to one another? Is the species that I've collected similar, different? So to what degree are they similar? To what degree are they different? Can I just use, for example, simple morphometrics to segregate species and come up with new species? I think one must be very careful about doing so. Before you go ahead and start interpreting any type of taxonomic data, you've got to be very careful about how reliable your phylogenetic studies are. What are the strengths in your phylogeny? What are the weaknesses that we had before? What are the different types of problems that taxonomists have raised before, and how I corrected them before embarking on a new project? What are the ambiguities that there are, for example, with species clustering in a particular clade, and you were not expecting that. Usually there are, unexpected results in phylogeny. How do we interpret that? Does your phylogeny result in a need to reevaluate the characters? Let's say if you're working on a particular group of species, especially plant pathogens, and you see that, there could be some information in terms of, those characters there, I mean, those morphological characters, or even in those molecular characters. Can I use them to try to convince, let's say, future plant pathologists about what needs to be done, what area or what segment of the DNA should we focus on? How are these important, for example, in terms of designing specific new primers to target specific pathogens? Are there any specific morph that has evolved and make those species under investigation somehow unique as compared to others? Let's say even if I'm describing one particular species, will others be able to describe that new species or that particular species precisely? Okay, so the art of fungal taxonomy, and it's important to promote biodiversity. Okay, so far what I've said is that limited emphasis is being placed on conservation biology when it comes to fungi. We tend to give priority to animals and maybe large microorganisms. We need a lot of data on host specificity. Are there really species that are host specific? Or fungi are potentially just opportunistic, and no matter what substrate you throw them there, they will just survive and thrive. What does biogeography have to tell us in terms of understanding a species diversity, species distribution, species gene flow? And how do we use all these to set priorities for conservation? I think there's a need to promote biodiversity. But for that, we need a coordinated efforts. We need to come in line with what others are doing. Example, with our fungal and culture collection protocols. We need to come up with a community-based conservation. There's a need for mycologists to engage more amongst themselves. And we need to appreciate that fungi is an important part of our nature. Okay, ladies and gentlemen, that was my last slide. I've been given 30 minutes. I've just given you a summary about fungi, a little bit on its importance in taxonomy, the use of DNA sequences as well in terms of establishing new species. I do hope that this has helped you a little bit about thinking in terms of possibly your studies as well. Thank you very much. I leave the floor open now and to Dr. Shanoy.
Wow, that was a masterclass on fungal taxonomy and fungal diversity. Thank you very much, Dr. Rajesh Jeewon, for inspiring us, for making us think critically about the issues. Many issues you have discussed. Probably we'll have to go back to the recording of your presentation and think about those. May I have the pleasure of asking you the first question? Dr. Jeevan?
Yes, sure, yes.
I mean, there are a couple of things I picked up from your slides. Species as a mental construct, and and in Yunnan you could see one new taxon out of 5 things you described there. Fine, that's interesting. But what I'm trying to ask you is actually a fungal taxonomic system or a classification system is expected to consider the following. First one is the statistical support or statistical soundness of the hypothesis or as the new species or new taxa you propose. Number 2, whether where your classification system would be used effectively without much hassle by the end user, for example, plant pathologists or the medical biology or medical microbiologist. The third one would be whether the developing a kind of a natural classification system for fungi, whether it is going to be cost effective. Can everybody afford the cost of developing it? And can everybody afford describing a new species using so many gene regions, um, um, or maybe whole genome sequence and hi-fi sophisticated tools? So what are your views on it, Dr. Jeevan?
Okay. I think Dr. Chanel, you've elaborated on a few aspects. Maybe if I understood, you wanted some comments on the last bit. Okay. To me, personally, to come up with a unified and standardized system that can meet the needs of most of these people there, it's very difficult. And you yourself, you pointed out that there could be plant pathologists and their strategies, their priorities are different. There could be others who are involved in biotechnology. Maybe to them, they will not be interested with your taxonomic framework. They will not be interested with your classification because all their priority would be of that particular species being a pure culture, and I can use it to get profit out of it in terms of, biopharmaceuticals and so on and so forth. Now, when it comes to taxonomy, there is something that you mentioned. It's about a natural classification. Yes, Dr. Chenol, we are proceeding to a certain extent on the right track since 1998, 1999, when we first started doing those kind of DNA work at the University of Hong Kong back there. I think then you joined later on for your PhD. You will see it has been gradual, and things are becoming a little bit more simple now and complex as well because of the wealth of information we are getting. So we should not try to confuse to a certain extent. So if you try to merge those two now, it will be difficult for us to come to a consensus right now. When you talk about your morphology, you talk about your DNA, it will be a little bit difficult. You also mentioned about the feasibility of doing things, about, multi-genes and so on and so forth. I raised the issue in my last slide about an integrated approach because I believe that there are things that were being done in a very wrong way. Some people just describing a particular species or sometimes even a new species based on a slight difference in morphology, based on slight difference in measurements. And to me, these could be subjects to different types of variation because the way that you see a particular species, I can see it in a different way. And if you're describing the same species, colonizing the same habitat, isolated from the same host, and using almost the same type of equipment and method, yet you're getting a different species and I'm getting a different species, I think to me doesn't make sense. So one of the major ways by which we can try to correct this is basically to move a unified platform where we try to use a polyphasic approach to double test, especially those kind of hypotheses that you mentioned. So is my species new? That's your hypothesis. Then you proceed ahead with your morphology, with your culture, with your DNA, okay? And then based on what you get, you propose something. That's where sometimes we need to go ahead, I believe, with our DNA, which should not be used in a way to, let's say, deter the use of morphology, but as a way to back up and provide more reliable information when our morphology starts to fail to a certain extent. Regarding feasibility, again, you mentioned whether many of us will not be able to use it. I think back there before year 2000, when I was doing my PhD, it was a bit difficult. It was very costly, even when we used to purchase all these stuff and so on. But I think now it's becoming quite routine now. and having a PCR in the lab, doing DNA sequencing, and getting the sequences, and with all those technology softwares that we have stuff online nowadays, I think it's opening the window and the door now for many people to embark more, especially Indian scientists, to embark more on DNA data to come up with more reliable classification scheme.
Can I just supplement with one more question, please?
Yes, sure, Dr.
Yeah,
Shanoy.
as we understand, a species is a hypothesis, and as I understand, hypothesis is also a mental construct. Okay, so
Yeah.
how do we differentiate a mental construct from a mental artifact?
Okay, this is what I raised in one of my slides. Let's say sometimes when you're examining a particular species under the microscope, or possibly when you isolate it, I think as a scientist, sometimes even I've seen a lot of students, as soon as they collect one particular sample, all they want to do is try to see whether it is a new species. I think that should not be our first priority. I think our first priority would be, can I accurately identify that particular species on that particular host and try to understand its ecological role? But the moment you start to think about having a new species and publishing it, then you're going against science to me. Because then you're framing your mind to a certain extent about, okay, what can I see to describe my species or the new species? And you will capitalize only on that 5% differences, whereby that 95% similarity with all other organisms on earth are being neglected. Sometimes some people asking me, oh, I've seen this difference in there, so my species is new. Then I would say, but you've got 98% similarity there, so your species, it should be that particular species. So it's apparently a way of thinking there. I think we need to go step back a little bit behind, okay, and then rethink about strategy about that particular species, come up with a very good rationale about our morphology. Should I consider, for example, slight differences in cultural variation as something important? If there are overlap in morphology, example in terms of maybe morphometrics, even if I'm using DNA, if there are slight differences in DNA, there could be a number of reasons as to why that fragment of DNA is slightly different from those existing one in gene bank. So there's a number of parameters to think about and work out before we come up with a proper rationale. And then we start to think about our species. So there is a need for a clear demarcation in here. It's understanding science and not in the pursuit of science for maybe something like publications and fame or whatever.
Thank
Thank
you
you.
so much. I will let Dr. Devdatta to ask you a question now. Thank you so much, Dr. Devdatta. Dr. Devdatta, please. Dr.
Sorry,
Devdatta?
sorry.
Yeah.
Yeah. It was nice talk, Professor Jeevan sir. Actually, I have one question. How does MALDI-TOF contribute to the current fungal classification? Because we do use MALDI-TOF in most of the diagnostics of pathogenic fungi in clinical cases. So, how we can think we can utilize this for current micro terrestrial fungi or fungi from diverse environments?
Okay. Thank you, Devata, for this question. Unfortunately, when it comes to those aspects you're talking about, it's still in a transitional stage. I remember when I was doing my PhD, I started to embark a little bit on proteomics, where these were the buzz these days back there, at 20, 21 years ago, when we started to think about, for example, you mentioned about pathogens. I was trying to look into and trying to elucidate to a certain extent how can I differentiate non-pathogenic organisms from pathogenic organisms. So could there be a proteomic profiles there? Could there be specific proteins that could help me demarcate between my species? So these were the birds at this point in time. But even now, after like 15 or 20 years, it's very difficult to come up with a proper schema or framework as to how we can try to integrate, for example, these chemicals, those secondary metabolized profiling that we're doing in our taxonomy. It's a bit far-fetched for the time being. Why I'm saying this is because those profiles that you are getting, we've got to understand that those profiles are highly subjective to a certain extent based on the living conditions that you have. So trying to tally one particular metabolite, one particular chemical, and try to link it with taxonomy, it's even more difficult than using DNA and link it with morphology and taxonomy. Even with DNA, we've got to bear in mind that with DNA, you're using a small proportion of the whole genomic information available, and you're trying to relate it to a species. And the same happens with a multi. When you're trying to get the profiles of some metabolites in there or of some chemicals and so on, and try to link it to one entire organism. So I think there's a number of precautions that we must need to take. But if the rationale in your work would be to try to elucidate whether these chemicals I'm getting is a function of specific working conditions in my in vitro work that I'm doing, and how can I optimize the experiment I'm doing for a particular metabolite output, then that's fine. But using it for the time being to come up with or link it with taxonomy, we should be very careful, because you've got to understand when we look at a particular organism, at their, let's say, phenotypes, we're looking at a vast area. So if we talk about, let's say, chemicals, should I be looking at all the chemicals that the organism is producing and try to identify and compare? The same happens with our genomic information. If we are using only one segment of our DNA and saying that, okay, yes, blah, 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 blah. But then the concept would be to use the whole genome sequence and try to get all the information in there. And then basically you can try to talk about functional genomics. You can try to elucidate everything, but this would take some time. I think someone has raised, I think this answers your question, Devadatta. I think Dr. Rajesh Kumar has raised his
Thank
hand
you.
as well. Maybe I'll leave the floor to Damodar
Chanoy,
as moderator.
can I ask a question?
Yes, Dr. Kumar.
Yeah. Yeah. My question is, you have emphasized the importance of morphology and integrated taxonomy in characterization of species.
Yeah.
In the view of recent developments, such as DNA as holotype and even genome-based taxonomy, that means recently even Aspergillus niger group, they have changed the multigene sequenced concept based on the whole genome based identification concept. I just want to have your opinion on these progresses and how we can tackle these challenging areas using the current tools which we are dependent upon for identification of the species.
This is excellent, Dr. Kumar. I think we've got to remember, not all species are the same, and not all species would behave in a similar way when it comes to fungal species. There are some species which actually model organisms, and we can use them no matter where. when you talk about Aspergillus, for example, it's a well-known species. People are characterizing them. It's a bit like when you talk about Cordyceps. You talk about some other famous Neurospora, for example. Now, when it comes to the application of, let's say, whole genome sequence, it's fine. But based on what we've learned over the last few years or so, we are still in a dilemma. We are still in a dilemma or undecided because the amount of information that we have from the genome sequence, how far it's contributing to taxonomy, it's setting us aside a little bit. Why? It's because we've got to understand to a certain extent is that we cannot compare the whole genome profile of one organism to another organism where we don't have such information. Even when it comes to a particular strand of DNA that you are using, when you're trying to use it and put it in a database like in GeneBank, we've got to understand the GeneBank, the repository there, the amount of sequences available is only a fragment of the world existing species. So if I'm getting my DNA there and blasting it and doing my analysis, I'm still comparing my samples with possibly a small fraction of the total number of species that is extant in the world. And then you're coming up with assumptions. So again, I said that these concepts are good to start with. It's not only for taxonomy, it's giving us a lot of other ideas, a lot of other avenues about functional genomics, about what could be the potential roles of those microorganisms in there as well, and how, for example, these different genes that we are working over can be used, for example, in terms of biotechnology. Yes.
Wow. Wow. Thank you so much, Dr. Rajesh Kumar and Dr. Rajesh Jeewan. We'll be continuing discussion when Dr. Jeewan will be visiting in India next year in February. Thank you so much, Dr. Jeewan.
Yes, and then possibly I can meet Dr. Kumar and Dr. Devadatta there.
Yeah, you will definitely meet Dr. Devadatta.
Thank you, sir. Thank you.
He's in Chandigarh, yes. Okay, I welcome to stay back and listen to my presentation, which will be for only 10 minutes because people are waiting for the kind of winding up of this whole lecture series. Thank you once again, Dr. Jeewan. Nice to hear you. Maybe I'll listen to you after 15 years. Maybe, I haven't met you for 15 years then, since 2007. Yeah, great. Thank you.
That's why I'm looking forward to come there next year. Let's hope we get to meet in Chandigarh next year.
Yeah, sure. Yeah, I would love. I mean, it has been a wonderful.
Sure, sir.
It has been a wonderful time listening to your ideas. Many things to agree, many things to disagree. Thank you so much, and for enlightening us and making us think critically for many of the issues. And I'll hand over this platform to Ramana Mirde, who is going to do something for me. Ramana.
Thank you, Rajesh sir, for such a wonderful lecture. I now welcome Dr. Bele Damodar Shinoy sir for his lecture. Before starting his lecture, I would like to give a brief introduction about him. Dr. Bele Damodar Shinoy is a well-known mycologist and principal scientist at CSIR National Institute of Oceanography Regional Center, Visakhapatnam. He is the managing editor of MycoAsia and MycoIndia journals. He has